Cells are the fundamental units of life. They're incredibly diverse, but they all share some common features. One of these features is the presence of internal compartments called organelles. These organelles are like tiny organs within the cell, each with its own specific function. To understand how cells work, we need to understand the structure and function of these organelles. One group of organelles is collectively known as the endomembrane system. This system includes the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. These organelles are all interconnected either directly or indirectly, and they work together to synthesize, modify, package, and transport molecules within the cell. Studying these tiny structures is a challenging task. We need specialized tools and techniques to visualize and analyze these organelles. Over the years, scientists have developed a range of powerful techniques that have revolutionized our understanding of the endomembrane system. These techniques include autoradiography, green fluorescent protein tagging, biochemical analysis, subcellular fractionation, cell-free systems, and the study of mutant phenotypes. These approaches have allowed us to piece together a detailed picture of how the endomembrane system functions. Autoradiography is a technique that allows us to track the movement of radioactive molecules within cells. This technique was instrumental in early studies of protein synthesis. Scientists used radioactive amino acids to label newly synthesized proteins. By tracking the movement of these radioactive proteins over time, scientists were able to map the pathway of protein synthesis. They discovered that proteins are synthesized on ribosomes located in the cytoplasm or on the endoplasmic reticulum. Autoradiography provided crucial evidence for the role of the endoplasmic reticulum in protein synthesis. It showed that proteins destined for secretion or insertion into membranes were synthesized on the endoplasmic reticulum and then transported to other organelles. This technique also helped to elucidate the role of the Golgi apparatus in protein modification and sorting. By following the movement of radioactive proteins, scientists observed that proteins moved from the endoplasmic reticulum to the Golgi apparatus, where they were further modified and sorted for delivery to their final destinations. Green Fluorescent Protein, or GFP, is a protein that emits green light when exposed to blue or ultraviolet light. This protein has revolutionized cell biology by allowing scientists to visualize proteins in living cells. By attaching GFP to a protein of interest, scientists can track the movement and localization of that protein in real time. This technique has been used to study a wide range of cellular processes, including protein trafficking, signal transduction, and cell division. GFP has been particularly useful for studying the endomembrane system. Scientists have used GFP to track the movement of proteins between organelles, providing further evidence for the interconnected nature of this system. For example, by tagging proteins involved in vesicle transport, researchers have been able to visualize the budding and fusion of vesicles as they move between different compartments of the endomembrane system. This has provided valuable insights into the mechanisms of protein transport within cells. Section 4. Cells under the microscope, visualizing the endomembrane system. The development of electron microscopy in the mid-20th century revolutionized our understanding of cell structure. For the first time, scientists were able to visualize the intricate details of organelles, including those of the endomembrane system. Electron microscopy revealed the complex network of membranes that make up the endoplasmic reticulum, the stacked cisternae of the Golgi apparatus, and the membrane-bound vesicles that transport molecules between these organelles. These images provided crucial evidence for the existence of these organelles and their roles in cellular processes. They also allowed scientists to develop more accurate models of the endomembrane system and its functions. However, electron microscopy has its limitations. It provides static snapshots of cells frozen in time. To study the dynamic processes of the endomembrane system, scientists have turned to other techniques such as live cell imaging. Section 5. Deconstructing cells, biochemical analysis and subcellular fractionation. While microscopy techniques allow us to visualize the endomembrane system, biochemical analysis and subcellular fractionation provide complementary information about its composition and function. Biochemical analysis involves breaking open cells and isolating specific organelles or molecules for further study. This approach has been used to identify the protein and lipid components of different organelles. Subcellular fractionation is a technique that separates different organelles based on their size and density. This involves centrifuging cell extracts at different speeds, resulting in the separation of organelles into distinct fractions. By analyzing the protein and lipid composition of each fraction, scientists can learn about the unique functions of different organelles. For example, they can identify enzymes involved in protein modification in the Golgi apparatus, or proteins involved in protein degradation in lysosomes. Combined with microscopy techniques, biochemical analysis and subcellular fractionation have provided a comprehensive understanding of the molecular composition and function of the endomembrane system.